Welcome everybody to this Royal Society of Tropical Medicine and Hygiene webinar on treatment optimization of major tropical infectious diseases with a focus on sub-Saharan African populations, the role of genetic variation and environmental factors. My name is Gail Davey. I'm Professor of Global Health Epidemiology at Brighton and Sussex Medical School, but today joining you from Addis Ababa University. This makes me extra delighted to introduce Professor Eleni Aklillu, who will deliver her Donald Mackay Medal webinar for us. Eleni is an outstanding Ethiopian tropical pharmacologist whose life exemplifies commitment to the Global South. Her research has centered around optimizing drug treatments for ethnically diverse Black African populations. Over the past 2 decades, she has investigated pharmacogenetic and environmental differences in drug metabolism and treatment outcomes in a range of communicable and non-communicable diseases in populations across sub-Saharan Africa. Her work focuses on major infectious diseases, including concomitant TB and HIV infections, HIV and malaria infections, and neglected tropical diseases too, specifically lymphatic filariasis, schistosomiasis, soil-transmitted helminths, and onchocerciasis. Capacity strengthening has been really central to Professor Eleni's work, and she's known not just as an innovative scientific leader, but as an inspirational mentor and an outstanding role model to young African scientists. Professor Eleni will speak for about 25 minutes, and after that we will have plenty of time for questions and answers. So please do post your questions in the Q&A function at the bottom of the Zoom window. And without further ado, I'm going to hand over to you, Eleni, to continue.
Very much, Professor Gail. I welcome the audience. I would like to thank the Royal Society of Tropical Medicine and Hygiene for giving me this prestigious award, the Donald Mackay Award. I'm very much truly honored and delighted to receive this medal award. And I acknowledge the contribution of my research group, everyone in my research group, my former PhD students and current collaborators from various institutions in Africa and in Europe. I also thank very much Professor Gail for noticing my work and nominating me for this award. In this presentation, I will give the highlights of our finding, which we have been doing for the past 2 decades in treatment of optimization and prevention of major tropical diseases, HIV, tuberculosis, malaria, and neglected tropical diseases. And our work was mainly focusing on identifying the risk factors for safety and efficacy of treatment of these infectious diseases. There are several factors contributing for treatment outcome and differences in pharmacokinetics of drug exposure. Biological factors include sex, age, and physiological factors, including pregnancy, and environmental factors includes drug treatment, concomitant medications, smoking, and chemical exposure. And the other one is genetic variations in drug metabolizing enzymes, transporter proteins, transcription factors, and drug receptors. Our research group from Karolinska Institute have done extensive research in identifying interethnic variations in drug metabolism capacity and treatment outcome. And we have identified really that there is a major difference between Black Africans, Asians, and white populations in terms of metabolic drug capacity. And generally, the Black Africans are relatively slow metabolizers compared to the white and the Asian population. And knowing this, I thought that probably the genetic makeup of the Black African population will have some impact in determining the treatment outcome of these infectious diseases. especially where there is concomitant medications and coinfections, comorbidity, nutrition, and genetic diversity may also have impact in treatment outcome of these infectious diseases. My genetic study started early in the 90s when I was doing my master's project for research at Karolinska Institute. That time I have done a genetic analysis or genotyping and phenotyping of an enzyme called CYP2D6. This is an enzyme which metabolizes several clinically important drugs, including codeine and antidepressant and antipsychotic drugs. And that this CYP2D6 is known to be genetically polymorphic with copy number variations, single nucleotide polymorphisms, and also there are presence of pseudogenes. In the white population, this enzyme has been extensively studied, and this study was the first one from Africa, I would say, that time, except with my colleague from Zimbabwe, who has also studied this CYP2D6 in Zimbabwean population. And the normal gene, having 2 copies of CYP2D6, gives about 29 kilobase in the restriction fragment length polymorphisms. But when I was doing my research that time, these extra bands were really puzzling because this was not normal. This shows that the gene is duplicated and multiduplicated. And I found one individual having 5 copies, 2 to 3 copies additional than the normal 2 copies of the variant, which means that those who have higher duplication of the enzyme means they are fast metabolizers and ultra-rapid metabolizers. That means the drug that is metabolized by CYP2D6 is eliminated very fast from the body. And the distribution of the CYP2D6 gene duplication is much higher, about 1/3 of the Ethiopians carry gene duplications, and which is also prevalent in the Middle East and Eastern Africa, and also the southern part of Europe. Those who have mutation, they are poor metabolizers. The dose has to be very much lower. These are the general populations. And those who are ultra-fast metabolizers, the dose of the drug has to be decreased. When we did this study in 1996, this genetic study was thought to be very much of not clinical use. But after about 20 years later, these studies that we have done in the 90s have been used as to revise treatment guidelines in Ethiopia. FDA has suggested to use CYP2D6 phenotype-based codeine use. Codeine is an anti-pain drug that is metabolized to morphine by CYP2D6. Those who are poor metabolizers are not experiencing so much of morphine, but those who are fast metabolizers, they have so much of morphine produced. And that means they are experiencing overdose of morphine. So FDA suggested that those who are fast metabolizers shall not use codeine. And also in children who are below 12 years of age shall not use codeine. And after an incident of fatal incident of a nursing infant whose breastfeeding mother was taking codeine and who happened to be ultra-rapid metabolizers, FDA also doesn't recommend to use codeine for breastfeeding women. So FDA of Ethiopia has used this input from our study that was done in the 90s to ban this drug because most of the Ethiopians are ultra-rapid metabolizers. When it comes to the infectious diseases, knowing that there is a huge inter-individual variation and between population variation drug metabolism, and HIV, malaria, and TB are very common in Black sub-Saharan African population. And these diseases are also using combination of drugs. That means there is drug-drug interaction, comorbidity, coinfection, and also genetic variation, which complicates treatment of these diseases. So it was really very important to do this study in sub-Saharan Africa population, in patient, in real world, to identify what are the risk factors for safety or toxicity outcomes of this treatment and how to optimize treatment of this co-infection in sub-Saharan African population. Our research group have conducted addressed several of these issues, concomitant medication of HIV and malaria, HIV and TB, and also the 3 drugs together, the 3 diseases together. The other question was that when to start antiretroviral drug in TB-HIV co-infected patients. As you know, TB drugs during the initial phase, there are 4 drugs to be taken. For HIV, there are 3 drugs to be taken. There is a lot of drug-drug interaction. If you start earlier, there is a risk of immune reconstitution syndrome, pill burden, and overlapping toxicity and drug-drug interaction. If we delay the drug, if we delay treatment of ART in TB-HIV co-infected patients, there is a risk of disease progression and high mortality. So because of the high mortality and also the complication when you start early, the WHO suggested to start ART during the first 8 weeks after starting TB treatment. But these are very critical because mortality is high during these times. So there was really a need to do research. There are several dilemmas in treating HIV in sub-Saharan Africa also, when to start ART in TB-HIV co-infected. Also, rifampicin is a potent inducer of CYP enzymes, which also are metabolizing antiretroviral drugs. And studies in white population and healthy volunteers, it showed that rifampicin reduces plasma concentration of Efavirenz by 1/3. Therefore, they suggested to give 600, instead of 600 milligrams, a standard dose to give 800 milligrams. But when Black African patients who have taking 800 milligrams have experienced a serious adverse event and toxicity from Efavirenz. And WHO didn't recommend 800 and was waiting for a evidence-based recommendation from researchers. But the Western countries, they were increasing dose of 800 milligrams. Immune reconstitution syndrome, adherence, concomitant medication, and also toxicity of Efavirenz that are CNS toxicity and liver toxicity was very much challenging. So our research group was addressing all these questions through the past 20 years. The first was that we conducted a clinical trial to identify when is the optimal time to start antiretroviral treatment in TB-HIV co-infected patients in Ethiopia. We randomized TB-HIV co-infected patients to start antiretroviral therapy after 1 week, after 4 weeks, and 8 weeks of starting TB therapy. And we followed the patients for over a year. And our primary endpoint was all-cause mortality. And secondary outcome was incidence of hepatotoxicity, TB iris, immunologic outcome, and virologic outcome, and a smear conversion rate. After a year of follow-up, we have reported that it is better to initiate antiretroviral therapy early for those who have CD4 less than 50 cells per microliter. At that time, it was recommended, ART was recommended only for those patients who have CD4 count less than 200 cells per microliter. And this was our finding indeed, which later we confirmed that there is improved survival if we start ART. What about the extent of drug interaction? Rifampicin is a potent inducer of CYP enzymes. And the CYP3A is an enzyme which metabolizes more than 50% of clinically used drugs. And to see how much of the extent of drug interaction is between rifampicin and Efavirenz. And for beta-hydroxycholesterol is an endogenous marker, plasma for beta-hydroxycholesterol is an endogenous marker to assess induction of enzymes by concomitant medication. And we conducted, these are control groups, HIV only patient taking Efavirenz-based ART only, and these are the TB-HIV co-infected patient who were randomized to start ART 4 weeks after rifampicin therapy and 8 weeks after rifampicin therapy. As you see here, we haven't found any significant difference between the two randomized TB patients. But of course, there is significant difference in the induction by rifampicin, but when to start ART really had no significant influence on CYP3 enzyme induction. So we concluded that rifampicin is a potent inducer, and when to start ART has no significant impact and no additive values that much compared to 4 or 8 weeks. And because of improved survival rate, we recommended it's better to start ART earlier, and there is no significant drug-drug interaction. So in this paper, we have provided pharmacological evidence to support ART initiation at early stage. And WHO recommended and revised the RT treatment guideline for TB-HIV co-infected and cited our paper in their recommendation. Our paper was one of those clinical trials which provided evidence for WHO to suggest, instead of waiting for 8 weeks to start ART, WHO suggested that it should be started within 2 weeks of initiating TB treatment, regardless of the CD4 count. So this was one of example where our finding has really helped in revising treatment guidelines. The other one is when to start, whether to increase the dose of Efavirenz when it is given with rifampicin. As I said earlier, studies in white and Asian population have shown that rifampicin is a potent inducer and reduces Efavirenz plasma concentration. So they suggested to increase the dose of Efavirenz from 600 to 800 milligram. To identify this, we conducted a parallel comparative study in those who are HIV only infected and HIV TB co-infected patients who are taking concomitant medication of Efavirenz plus rifampicin-based anti-TB therapy. And surprisingly, we have found inconsistently higher Efavirenz plasma concentration than those Efavirenz only treated patients. This was extremely different and which is completely opposite from what was expected. That's why we need this kind of studies in our population in sub-Saharan Africa, in real patients, instead of just extrapolating findings from the Western society. It's better to always test the treatment guidelines and do base our recommendation based on findings from the local population. It was very difficult for us to publish this paper at the beginning because it was very much unexpected, but we have confirmed a similar study doing in Tanzania and Uganda population. We found that either rifampicin has no significant effect on Efavirenz plasma exposure or higher in the case of Ethiopians. And we found no immunological differences and biological differences in the treatment of the two groups. So we suggested that there is no need to increase the dose of Efavirenz when given with rifampicin. This is an extensive study that we did in Ugandan population. We have monitored the plasma concentration of Efavirenz over almost a year period, and we found no significant difference. Even after stopping TB treatment, we followed them and the Efavirenz concentration was not significantly different. Then we performed pharmacokinetics and pharmacodynamics modeling and simulation of our data and suggested that even 600 milligram Efavirenz dose is much higher than necessarily. So we suggested a lower dose of Efavirenz for the general population, or even much lower those who have mutation is homozygous mutated for the CYP2B6 enzyme which metabolizes Efavirenz. This is a study from HIV-only patient. And in another TB-HIV co-infected patient, we also confirmed that rifampicin is not a significant predictor of Efavirenz exposure. And still in TB-HIV co-infected patient, we recommended a lower dose of Efavirenz. And here this shows that there is no correlation whatsoever between the viral load decay, which was followed over a year, versus trough concentration of the plasma concentration of Efavirenz. This lack of correlation shows that Efavirenz plasma concentration is way high. Probably it's not really predicting treatment outcome. This our low dose suggestion of Efavirenz have been confirmed by other groups clinical trial who showed that 400 milligram Efavirenz is much better in terms of safety outcome compared to the standard 600 milligram dose. And later, WHO updated its treatment guideline in 2019 and suggested that low dose Efavirenz in combination with NRTI backbones to be an alternative first-line drug regimen. And our study really now in this case has contributed for treatment revision. In another, we have conducted a similar study in East African population, in Tanzania, Ethiopians, and Uganda. So why did we do that? Because the sub-Saharan African population is so genetically diverse. One study outcome from Ethiopia cannot be extrapolated directly to the neighboring country, let alone to the entire continent from Tanzania and Ethiopia. We have found significant differences, having the same treatment and the same type of diagnosis or HIV treatment. there is significant differences in the plasma exposure between Tanzanians and Ethiopia. Plasma exposure of efavirenz is much higher in Tanzanians than Ethiopians. We found also significantly different genetic variation between the two populations. And these are the two PhD students from Tanzania and Ethiopia who did this study. Not only on efavirenz plasma exposure, but also on treatment outcome in terms of liver enzyme abnormality, there was between HIV only and TB in HIV patient in Tanzanians was much significantly lower than the Ethiopians. The Ethiopians, despite of having lower efavirenz plasma exposure, they have much higher incidence of liver enzyme abnormality compared to the Tanzanians. This shows that between population variation and treatment outcome and the need for population-specific studies. We also identified that this CYP2B6*6 mutation is a marker for liver enzyme abnormality. Those who have developed liver enzyme abnormality have a much higher exposure of plasma compared to those who haven't developed drug-induced liver injury. And this is a hazard, cumulative hazard between those who are mutated, homozygous mutated, compared to those who have functional variant alleles of the CYP2B6. We also identified a similar variant allele to be a marker for neuropsychiatric manifestations. This study was in Ugandan population, where we have followed TB and HIV co-infected patients receiving efavirenz, and we monitored the efavirenz plasma concentration and also CNS toxicity, associated CNS toxicity. It shows that genetics really plays a role, both for CNS toxicity and liver toxicity. While WHO is revising and recommending low dose efavirenz for adult population, it's still not recommending it for pregnant women because there is no data, and low dose efavirenz is not still recommended for children. So currently we are conducting an RSTMH-funded clinical trial in Uganda and Ethiopia comparing efavirenz 600 mg and dolutegravir versus low efavirenz 400 mg in pregnant women, HIV infected pregnant women. And we follow this pregnant woman over during the pregnancy and after the pregnancy, and our primary outcome is PMTCT, prevention of mother-to-child transmission of HIV, and viral and immunological outcome. This study is ongoing, and this is our website if you are interested to see more. We conducted also a clinical trial to identify optimal treatment regimen for prevention of malaria during pregnancy. In malaria high transmission areas, pregnant women during pregnancy, they are receiving sulfadoxine parametamine monthly dose during pregnancy to prevent malaria. However, sulfadoxine parametamine is no more used to treat malaria because of parasite resistance, but still sulfadoxine parametamine is safe during pregnancy and it is used. However, because of this high parasite resistance, we suggested that there might be better to look for another intermittent preventive treatment than sulfadoxine parametamine. In this clinical trial, we conducted comparative study among pregnant women who were randomized to receive dihydroartemisinin piperaquine, which is an artemisinin combination therapy, versus the standard sulfadoxine parametamine. And about 1,000 pregnant women, malaria-free, during the antenatal care visit, they were randomized to receive these two regimens, and we followed them during the entire pregnancy period. And our primary outcome was placental malaria, histopathologically confirmed placental malaria. And our secondary outcome was overall malaria at delivery, symptomatic malaria, parasitemia during pregnancy, and adverse birth outcome in terms of low birth weight, stillbirth, abortion, and also any adverse outcome. Our study has shown that there is a higher protective efficacy of dihydroartemisinin piperaquine compared to the standard sulfadoxine parametamine. This is a hazard risk of having malaria during the intermittent preventive treatment. In this study, the PhD student was really on call and followed at delivery time, or at delivery what to call it, placental tissue, and for microscopy, and he conducted cord blood, placental peripheral blood to look for PCR for malaria detection. And this data has been published and also shared with Malaria in Pregnancy Consortium and is now in consultation with WHO if there is a need to revise IPTP for malaria prevention. And we conducted also another randomized clinical trial comparing two treatment regimens to treat schistosomiasis, intestinal schistosomiasis. As you know, praziquantel is an old drug that has been used for more than 40 years, but still schistosomiasis is a problem. Children are receiving annually as mass drug administration, and still schistosomiasis is a burden and the problem. This is partly because that this drug is effective in killing the adult worm, but it's less effective against the juvenile or immature worm. In vitro studies have shown that dihydroartemisinin piperaquine is killing the immature worm. So in this clinical trial, our hypothesis was that by targeting both the mature and immature parasite, developmental stage of the parasite, if we combine these two drugs compared to the praziquantel, the standard one, we will get a better efficacy. Our primary outcome was a cure rate and egg reduction rate after 3 weeks of treatment and 8 weeks of treatment. So we found a superior effect of praziquantel and dihydroartemisinin. Combination therapy is much effective than praziquantel alone. And this, the adult worm, after 3 weeks of treatment, we normally expect that there wouldn't be no significant difference between the two because the mature schistosome requires like 3 weeks of time to get adult and mature and start to lay egg. But this was really surprising. And we wanted to know why this is the case. And we conducted a drug-drug interaction between the two combination drug. And those who have been receiving combination therapy have a higher plasma exposure of praziquantel, both praziquantel, the enantiomer R praziquantel and S praziquantel. So this is really very helpful. And if this study is replicated, it will again be a game change for schistosomiasis elimination and control. Recently, we are working on pharmacovigilance capacity building and strengthening in East African population. As you know, there is an increased access to medicine and number of clinical trials being done in Africa is higher, and the medicine regulatory authorities are the one who are monitoring clinical trials and regulating medicines. However, there is a limited capacity for pharmacovigilance. And this Proforma project is a 5-year, RSTP funded and co-funded by SIDA, a project to strengthen pharmacovigilance in Ethiopia, Kenya, Tanzania, and Rwanda. And also there is an increased mass drug administration and vaccination. It's an example for COVID that mass immunization is also ongoing. But however, there is limited stakeholder coordination between public health program, regulatory authorities, and the academia. The Proforma partnership is a combination, a partnership between academia for sustainability and between medicine regulatory agencies who are training to change policy into practice, and the stakeholders are public health program, regional center of excellences for regulation, and WHO collaborating centers. And Proforma has established really this collaboration using each of these 4 participating East African countries. And jointly we conducted active safety cohort event monitoring in Tanzania and Kenya. The local investigators selected to do mass drug administration for lymphatic filariasis, where we have monitored the safety and efficacy of single dose administration of ivermectin and albendazole in Tanzania and Kenya, triple versus dual therapy for lymphatic filariasis. And more than 20 individuals have been monitored for safety after receiving mass drug administration, and the result is being considered for application. In Tanzania and Ethiopia, we also conducted a training for HPV vaccine surveillance. And in Ethiopia, we have conducted vaccine safety surveillance. And that experience gained from Ethiopia has been used really for COVID vaccine surveillance in the country. In Rwanda and Ethiopia, the investigators selected to study mass drug administration for schistosomiasis and soil transmitted elements. And there are 5 PhD students are working on this active safety and efficacy cohort event monitoring in these 4 countries. Not only we are doing active safety surveillance, but also we human capacity building is our pillar of our project. We have developed short courses for pharmacovigilance, like risk management and communication and signal detection and reporting through the Proforma project, which is led by our partner from Lariv, the Netherlands. And we have 7 PhD students are being trained, and 5 master students are being trained. We have developed undergraduate and postgraduate PV curriculum, which is now being implemented in the medical universities in our participating countries. We have developed also e-learning module for healthcare professionals as in-service and pre-service for healthcare professionals to refreshen their pharmacovigilance capacity and practice. A number of PhD students have graduated from this PhD project. Our project have created opportunity for African young researchers to get their PhD. And these are 9 PhD students who were registered at Karolinska and graduated. And these are the ongoing PhD students now who are going to graduate next year. Our general director from the Tanzania Medicine Drug Authority, and Joseph Kabatende from Rwanda, our Abby from Gambia, and Tigist and Christabel from Kenya. They are currently doing their study. These are just to share experience, our field experience from Ethiopia. This was a workshop for HPV vaccine surveillance. And this was schistosomiasis vaccine surveillance, schistosomiasis MD surveillance. And we have conducted data training for school teachers and also dispatched lab technicians to collect stool sample for schistosomiasis and cell transmitted elements detection. This is a Rwandan experience where we have done our study in 4 districts along the Lake QV. And this is an explanation of our study for the students, the children, school children who participated in our study, the school teachers, and also this was a meeting with parents. And some of the study sites were really very remote and there was no access, road access. And these are our PhD student study coordinators traveling with wooden boat to our study site. So this is an experience from Kenya where we have done lymphatic filariasis mass drug administration safety surveillance where lymphatic filariasis is much common in Mombasa. This was meeting with the communities, and these are community leaders and religious leaders who were very much positive and supportive, and these are community drug distributors who were trained on data collection. And this was a meeting in our hotel after a field work. This was experience from Tanga, where lymphatic filariasis is much common in Tanzania. This was a rural area. We have conducted this study, and we screened for antigenemia, for blood samples were taken, and we conducted night blood screening for lymphatic filariasis. And this is our student who was doing microscopy. I would like to acknowledge my collaborators from different institutions, Professor Yasu from Addis Ababa University, Omari Minzi Apollinary from Muhimbili University, Professor Emily from Rwanda, and the list goes on, and Dr. Jackson Kunzo from Uganda, Dr. Birganet Lamun from Hawassa University, and my European collaborators, Professor Alamuddin Zumla, Eugene Stefano from Italy, and the list goes on. If I don't mention you here, please consider that you are in my heart. And thank you so much for the wonderful work that we have been doing together. And I would like to acknowledge also the Proforma and the PREGARD consortium who are currently ongoing. And I would like to acknowledge our funding organizations, EDCTP and SIDA and the Swedish Research Council for funding this project. And thank you very much for listening.
Thank you so much, Eleni. That was really wonderful to get that overview of a couple of decades of work. It's really inspiring to hear about it all put together in that way. So many thanks indeed. And there are a couple of questions that have come in, and I'm going to just encourage others to put questions into the Q&A, and I'm going to give you a little bit of time to do that, because I'm actually going to take chair's privilege and ask Professor Eleni a question of my own to start. And that came up quite near the beginning of your talk, when you talked about the time it took from understanding the fast metabolizers and the change to regulations or the change to treatment guidelines. And I suppose I just really love to hear your reflection on what helps policymakers to take up and use and change guidelines most from us as scientists. What's your view on that?
It's a very good question, Gail. It's really impressive that time when we were doing genetic studies in the 90s, it was considered as not clinically relevant for sub-Saharan African population. And even some of the reviewers suggested that, okay, you have done a fantastic genetic study, but what is the use for the patient care? But my answer to the reviewer was that science shall not be based on availability of resource and finance. And our task as a scientist is to generate knowledge, and policymakers will make use of it after a long time. But African regulators, they have limited capacity and ability to utilize this kind of research. And had it not been the US FDA citing my research paper and the incident of codeine adverse event, which was shown to be fatal, and a warning given by US FDA was just copied by the Ethiopian National FDA to make this warning. So there is a huge gap in using the data generated from sub-Saharan Africa, from the community, to make it useful to revise treatment guidelines. Of course, WHO waits for a longer time, but the national regulatory authorities should make really use of the data that's generated in their own population for the national use, in-country use. And it took almost 20 years to cite that paper. Even that time, when the National FDA of Ethiopia cited the US FDA, they didn't know that this study was done by an Ethiopian. It was later that they found out.
Interesting.
So, of course, how to present our scientific work for the policymakers in a language which is understandable is also another part which we should work on, so that it can be really usable and understandable by the politicians, by regulators, to make difference in the treatment guideline.
Thanks. Yes, I do agree. There's some responsibility that we must take to present in a way that's really appropriate to each audience. So yes. Many thanks for that answer. Now, you've got lots of sort of very positive comments there in the Q&A, and I'm going to take a question now from Colette Dandara, and they're saying interesting work. The differences in EFE exposure between Tanzania and Ethiopians. Is there any role for rare pharmacogene variants in the spectrum of genes affecting EFE disposition?
Yes, I know Colette. He's a colleague of mine. He's one of the pharmacogeneticists from Africa, but we have done a lot of work from South African and West African population. And indeed, that CYP2B6 genotyping is a marker for adverse event associated with treatment of Efavirenz. And there is really a potential to use genotype-based dose modification, which we have recommended in our publication from Uganda. And at the moment, we are not at a situation where to use genotype-guided prescription of doses, but I'm sure this will be improving, and there is a possibility in the near future. It may not take like 20 years as it has taken for codeine and CYP2B6 genotyping, but I'm sure that in the near future, the genotype will have some impact and use for patient care and the clinic use.
Great, many thanks. And then there's a question, that's almost echoed by two people here, alongside several more very positive comments. And so I'm going to read one from Temesgen, who would like to ask you about the possibility of making pharmacogenomics accessible in African institutions. So the comment is that irrespective of inter-individual variability, there's rare access to genomic or genetic studies which might impact the pharmacotherapy of drugs. So it's a sort of accessibility question here. Yes,
nowadays there are companies who are working on bedside genotyping testing, which can be really used at the clinics. For example, in Europe, in Sweden, we are doing genotyping for cancer patients who are taking some anti-cancer drugs, where those who have this mutation are at high risk of adverse event. So those who have this mutation are either receive a lower dose or change another treatment for instead of using the standard dose. And this kind of possibility may be happening in Africa, but in rural areas where we have limited capacity to use, it may take some time, as I said earlier, but it's not far away. Nowadays, the technology, the diagnosis for genotyping has become really advanced. And in the near future, we may be able to use pharmacogenetics application. But at least we know that the genetic diversity in African population is so important to predict treatment outcome. Even it can be used for new therapeutics and identify susceptible genes for diseases and also toxicity, treatment-associated toxicities. So this is really an eye-opening what we are doing, and genetic research in Africa is flourishing through H3 Africa, and there are a lot of really genius geneticists coming, although we are one of the pioneers in the field, but I'm sure it will be make use of in the future. But at least the outcome that we are doing is really very important to revise treatment guidelines, and we are not really we shall not use a universal one dose fits all principle, and treatment guideline varies between should vary between population.
Thanks. Now I'm going to do two very quick further questions. There are also questions about whether the PowerPoint might be shared, which I'll leave to the RSTMH team. Colette again says, could you comment on how the graduated students continue to get support to continue with their research? And then I'll give you one more question after that.
Yeah, this is a very good question. And I'm very proud to say that our PhD students are really, really good. And some of them have already established their own research group. For example, Dr. Jackson Mogonzo from Uganda, and Dr. Birkene, my former PhD student from Hawassa University, has jointly applied and found a grant from EDCTP to do a clinical trial, which I am also collaborating with them. So I have established, sustained this collaboration with my former PhD students, and some of them are serving in the US FDA, and one is also working in International Vaccine Institute. Of course, even if they are spread around the globe, nowadays where you are is not a limit, and we are collaborating together.
That's a really lovely story of you now collaborating with former students. That's wonderful to hear. Really good. And I'm just going to take one more sort of more content question from Kirubel Hailu, and they ask, as you know, in Ethiopia and Kenya, we have a relatively high incidence of HIV serodiscordant couples. Does your group have any work or genetic explanation for that? And my sense is that probably is a slightly aside issue to what your group is doing, but please do answer if you've got any light to shed on that.
Yes, this HIV discordant have been really a lot of investigation on that, and some genetic makeup may be preventive. There is one study which was published earlier from Germany, who have an HIV patient who have received a bone marrow transplantation, became HIV free after that. And there are some identified markers for that. And that mutation, for example, this was a mutation in HLA gene. induction, maybe it's very rare, it may be protective. But since I haven't done any study in this area, I cannot say much. It may play a role, genetic may play a role in preventing this susceptibility to diseases. Is really, genetic plays a big role on that.
Thanks very much. Now, there are some other interesting questions there, but I feel we should wrap up now. We're meant to finish at 1:45. So again, I just want to thank you so much, Eleni, for sharing that rich experience. And I think everybody could see, you know, this wonderful capacity strengthening approach that you've applied throughout, and that there's a lot of comments about that in the Q&A as well. So many thanks. And to all our audience, thank you for all those questions and the comments too. I would like to thank you for joining us for this Donald Mackay Medal webinar. Thank you for participating in the Royal Society of Tropical Medicine and Hygiene's activities like this, and I do hope that you use the Society in many other ways, use a membership area and attend other meetings in the future. You will be very welcome. But for now, I'm going to draw the meeting to a close. You will be receiving a comment form after the webinar, and it would be great to hear any feedback that you have. So thanks all again very much for attending today, and congratulations again to our Donald Mackay Medal recipient, Professor Eleni Aklillu. Goodbye for now.
Thank you. Bye.